Welcome to the deep dive. Today, think about some really critical things we use, maybe injectable medicines or even just sterile eye drops.
Yeah, things were clean isn't nearly enough.
Exactly. They have this non-negotiable quality, don't they? They have to be absolutely free of any living microorganisms. Completely.
That's right. And that's what we're diving into today, the science behind achieving sterility. It's way more complex than just, wiping something down.
So our mission here is to really unpack the methods, the controls, the critical checks, the validations that make these products safe for you.
And we've looked at quite a bit of material microbiology sources, sterilization guides.
Yeah, and the first thing that jumped out, for me anyway, was that sterility, it isn't like a 100% guarantee for every single vial or bottle off the line.
No, that's key. It's actually probability, a very, very carefully calculated probability.
A probability. That kind of changes how you think about it, doesn't it?
Yeah.
Especially for something like an injection. It really shows you how big the challenge is.
It does. So maybe let's start with the basics. What is sterility, technically?
Good point. Lay it out for us.
Well, in this context, it means the complete absence of viable microorganisms, living ones, basically.
Okay, complete absence. But then you mentioned probability.
Right, because the benchmark we use is something called the Sterility Assurance Level, or SO.
SAL, okay, and I saw a specific number mentioned everywhere. 10 overall. 10 to the -6. What does that mean in, plain English?
It means the probability of finding a non-sterile item should be no more than 1 in 1 million. 1 in a million finished products.
1 in a million. So it's not saying every single item is sterile, guaranteed. It's about managing the risk down to incredibly low odds.
Exactly. Because, and this is fundamental, achieving absolute demonstrable sterility for any single item after it's gone through the process, well, you can't actually guarantee it or prove it for that one item.
Wow. Okay.
Okay.
That feels counterintuitive somehow.
Yeah.
So where is this level, this 1 in a million target? Absolutely essential.
Well, it's critical for a whole range of things. Parenteral products, that means injectables, ophthalmic stuff like eye drops.
Ah, right. Things going into the body or sensitive areas.
Precisely. Also, intramammary treatments, some inhalations, irrigation solutions used in surgery, intrauterine preparations, and even some things for severely injured skin, like certain creams or ointments.
So because you can't guarantee it for one specific item, that leads us straight to validation. Is that the idea?
Exactly. Validation becomes absolutely crucial. You have to trust the process because you can't test every single item to destruction.
Makes sense. So what makes a sterilization process effective? What needs to be right for it to work?
Several things. The process itself, obviously.
Yeah.
Then the conditions, time, temperature, moisture levels. Crucially, what microbes are already there before you start, that's called the bioburden.
Bioburden, okay, the starting level of contamination.
Yep. And even the product formulation matters. Its water content, pH, maybe it has some antimicrobial stuff in it already. And a key point is that killing microbes, it follows an exponential curve.
Meaning?
Meaning you kill a certain percentage in a given time, then the same percentage of what's left, and so on. So mathematically, you never quite reach absolute zero. There's always a vanishingly small, non-zero probability of a survivor.
Like trying to get every last grain of sand off a beach.
Kind of, yeah. You can get incredibly close, but guaranteeing absolute zero is, well, it's theoretically impossible.
Okay, so if there's always that tiny chance, we need a really robust way to be sure the process works. You mentioned validation. That's the key.
That's the unsung hero, yes. Validation.
So tell us why it's so critical. What does validation actually do?
It means every single sterilization procedure you plan to use has to be studied really thoroughly. You look at its effect on the product, the container, make sure it actually works in that specific case, and that it doesn't damage anything.
Right. Sterile but ruined isn't helpful.
Exactly. And then it has to be formally validated before you use it for real products. If you don't follow a validated process to the letter, you risk shipping non-sterile products or products that have degraded. Big risks.
And I guess the container is part of that validation too.
Oh, definitely. The containers, the closures, like the rubber stopper and cap, they're chosen carefully. They have to allow the sterilization method to work, maybe let steam heat penetrate, and they have to keep the product sterile for its whole shelf life.
Makes sense. And I read something interesting about parametric release. What's that?
Ah, yes. That's fascinating. For some well-understood terminal sterilization methods, that means sterilizing the product in its final container, like steam, dry heat, radiation if the process is fully validated and monitored.
Ah,
Regulators might allow a batch to be released based only on the process data, temperature logs, pressure readings, radiation dose, etc. No end product sterility testing needed for that batch.
wow. So the process data is the proof. That shows incredible confidence in the validation, doesn't it?
It really does. It means the validation was so thorough and the routine monitoring is so good that the physical process data guarantees the outcome, but that requires a lot of work up front.
I bet. So you sterilize it, it's validated, maybe even parametrically released, but then you have to keep it sterile, right? Prevent recontamination.
Absolutely vital. All that effort is wasted if contamination gets in afterwards. So strict precautions are needed after the sterilization step. Controlled environments, careful handling.
And this is where biological indicators come in.
GBI.
Exactly. They're another tool in the validation toolbox.
So what are these BIs? Like little test bugs?
Pretty much. They're standardized preparations of specific microorganisms, usually tough bacterial spores, known to be highly resistant to the specific sterilization process you're testing.
Spores, right? They're notoriously hard to kill.
They are. So these BIs provide a defined, known challenge to the process. You use them mainly during the development and validation stages, not usually for routine monitoring, unless it's specifically required for a certain process.
So they're like a stress test. How do you use them? Stick them in with the product. Essentially, yes. You might use them under slightly reduced sterilization conditions during validation. The idea is to show that even under these weaker conditions, where maybe a few of the BI spores might survive, it proves the BI is genuinely resistant. Then, when you run the full validated routine cycle, you expect complete kill, no survivors.
Okay, so you prove they can survive a weak cycle to show they're tough, then confirm they don't survive the real cycle.
You got it. And selecting the right BI is crucial. It depends on the method, heat, gas, radiation, how effective the process is supposed to be, the exact conditions, and the product itself, and you always place them in the hardest to sterilize spot in the load.
The cold spot or the place a gas reaches last.
Exactly, the worst case location.
Makes perfect sense. Okay, let's get into the methods themselves. First up, the classic. Steam sterilization. Moist heat. How does that work?
So steam sterilization, usually in an autoclave, which is basically a pressure cooker, it works by condensing steam. When saturated steam hits a cooler surface, it condenses, releasing a lot of latent heat very efficiently.
Right, that phase change releases energy.
A lot of energy. And for items directly exposed, the moisture itself also helps kill microbes. But you need full steam penetration, no trapped air. For liquids in sealed containers, like vials, the steam heats the outside of the vial and that heat conducts through to sterilize the liquid inside. The chamber acts like a steam jacket. Sometimes steam-air mixtures or even superheated water sprays are used for closed containers too.
And is there a particular challenge with liquids in sealed containers, like pressure buildup?
Ah, yes, very important. You often need air overpressure in the chamber during the cycle, especially during cooling,
Why is that?
to counteract the pressure building up inside the container as the liquid heats up. It minimizes the pressure difference across the container wall. Without it, especially with things like pre-filled syringes or some plastic containers, they could deform or even burst.
Ouch. Right, that seems like a critical detail. Is there a standard cycle?
The classic reference cycle is 121 degrees Celsius for 15 minutes, using saturated steam. But the minimum acceptable temperature is generally 110 degrees C. We also talk about F value.
F0, what's that?
It's a way to quantify the total killing effect of the cycle, kind of like a lethality score expressed as equivalent minutes at 121 degrees C. Even if the temperature fluctuates a bit, you can calculate the total FU. The minimum acceptable FUO and the slowest heating part of the load is usually 8 minutes.
Okay, a standardized measure of kill power and routine checks.
Monitoring the camera pressure and temperature profiles every cycle, especially in that cold spot, and calculating and recording the F if the system allows, it's all about consistency.
Okay, so steam is the go-to for things that can handle heat and moisture. What if they can't? What's the dry option?
That would be dry heat sterilization, still using heat but without the moisture. It's also a terminal method, usually done in special ovens with forced air circulation or sometimes in tunnels.
Hot air, basically.
Yeah.
How hot and for how long?
A typical reference cycle is 160 degrees Celsius for at least 2 hours. But again, the cycle has to be validated to achieve that SL of 10 occ or less. Validation uses temperature mapping, finding the cold spot, and often BI is designed for dry heat.
I heard dry heat is also used for something else. Depyrogenation. Is that sterilizing?
It's related but distinct. Depyrogenation uses much higher dry heat temperatures, usually above 220 degrees C. Its goal isn't primarily to kill microbes, but to destroy pyrogens, especially bacterial endotoxins, bits of dead bacteria that can cause fever if injected.
Ah, so it's cleaning up the bacterial debris, not just the live ones. Crucial for glassware like vials, right?
Exactly, for vials, equipment parts. Validation here focuses on showing you get at least a 3-log reduction, a 1,000-fold decrease in endotoxin levels. You don't typically need BIs for this, you measure the endotoxin itself. Routine control is monitoring the temperature in the coldest spot.
Got it. Okay, moving on. Ionizing radiation. Sounds intense.
It is quite powerful. This uses gamma rays, often from cobalt-60 sources, or high-energy electron beams, sometimes X-rays. The radiation damages microbial DNA and other vital molecules.
And what do you use it for?
Lots of things. Terminal sterilization of finished products, especially single-use medical devices. Also for inactivating microbes in tissues or cell-based products, sterilizing containers for aseptic filling. Low-energy electrons can even just sterilize surfaces.
Is there a standard dose, like the temperature for heat?
Yes, the typical reference absorbed dose is 25 kilograms, 25 kilograms. It's a measure of the radiation energy absorbed. And like the others, it needs validation to prove it achieves SAL 10 BIs might be used sometimes, depending on the product. Routine control is all about dosimetry, carefully measuring the delivered radiation dose to ensure it hits the target every time.
Okay. Next up, gas sterilization. Or vapor phase sterilization. How does this one work?
This is mainly for surfaces, for things that can't take heat or maybe radiation. Think primary packaging materials, parts of equipment, maybe some sensitive drug substances. The gas has to penetrate, react, and then crucially be removed.
Why is removal so crucial?
Because the gases used are often pretty nasty. There are two main types: alkylating agents like ethylene oxide or ETO. Big issue with ETO is that it's toxic, carcinogenic, mutagenic. So getting rid of residues after sterilization is paramount for safety.
Yikes. Okay. What's the other type?
Oxidizing agents, things like hydrogen peroxide vapor or peracetic acid. These are also highly reactive and toxic, so again, removal is key. The process usually has phases: conditioning, the actual sterilization exposure, and then aeration or degassing to remove the residues.
And what affects how well it works?
Gas concentration, temperature, humidity, moisture is often critical for gas sterilization exposure time, how the load is configured, the product itself, the packaging, lots of factors. Routine control involves monitoring all those cycle parameters and usually using specific BIs resistant to the gas.
Okay, so far all these methods kill the microbes. But you mentioned one that doesn't. Filtration. How does that achieve sterility?
Right. Membrane filtration is unique. It removes microorganisms physically rather than killing them. It's used for liquids and gases that just can't tolerate heat, radiation, or gas.
Like filtering water, but on a microscopic scale.
Exactly. Using a filter membrane with incredibly small pores, typically 0.22 micrometers or milliliter nominal pore size. That's small enough to trap most bacteria. The filter works by sieving, but also by adsorption microbes sticking to the filter material.
Just the pore size? Or is it more complicated?
Oh, it's more complicated. Effectiveness depends on the filter itself, its structure, surface chemistry, but also how it interacts with a specific product, the pressure used, the flow rate, how long you filter for. It needs very careful product-specific validation.
And the filter itself needs to be sterile, I assume.
Yeah, absolutely. The filter unit is either sterilized beforehand and assembled aseptically, or sterilized in line as part of the system. And routine control involves checking the bioburden before filtering, doing filter integrity tests before and after, use like a bubble point test, monitoring the process duration, volume filtered, pressure. It's all about ensuring the filter performed as expected.
Okay, one last method, or maybe it's more of a strategy. Aseptic assembly. What's that about?
This is different again. It's not a sterilization method itself. It's about maintaining sterility when you assemble a product from components that have already been sterilized using other methods.
Ah, like putting together a sterile drug vial. Sterile vial, sterile stopper, sterile drug solution, all done in a sterile environment.
Exactly that. Aseptic filling is a classic example, but also freeze-drying sterile products, aseptic blending of sterile powders, aseptic packaging.
What's the focus for validating this? It sounds like the environment and the people are key.
They are absolutely critical. Validation focuses on the clean room environment, personnel training and gowning, how critical surfaces are sterilized, how the pre-sterilized containers and closures are handled and transferred without contamination, and even how long the sterile product can be held before it's finally filled and sealed.
How do you test if it's working? You can't easily see contamination.
Process simulations are key. They're often called media fills. Instead of filling with the actual drug, you fill containers with a sterile microbiological growth medium like tryptic soy broth. Then you incubate the sealed containers.
And if anything grows.
It means contamination got in somewhere during the simulation. It's a direct test of the entire aseptic process, including personnel and environment. That, plus regular sterility testing on actual product batches, provides the assurance.
This has been incredibly thorough. We've covered a huge range of methods. But you mentioned zooming in on one. Moist heat for liquids. Why the extra focus there?
Well, it's just so common for injectables, IV fluids, things like that. And there are some interesting nuances in how it's validated, especially when the product is a bit sensitive to heat.
Right, you mentioned the overkill method versus the bioburden biological indicator, or BBBI method. Let's revisit that. Overkill is just hitting it really hard with heat.
Pretty much. If the product can take it, you use conditions that provide a massive safety margin far beyond what's needed to kill the expected bioburden. It's simpler to validate.
But if the product can't take it, like a sensitive protein drug,
Then you have to use the BBBI method. And this requires really knowing your bioburden, what types of microbes are typically present before sterilization, how many there are, and how resistant they are to heat.
okay, so you need detailed homework on the starting contamination.
You do. And then the aha moment, as you called it earlier, is how the BI is used here. Remember, the BI spores are chosen to be much more resistant to heat than the typical bioburden.
Right, the superhero bacteria versus the everyday microbes.
Exactly. So in the BPVI validation, you might run cycles where you demonstrate only partial kill, or maybe kill of just a moderate number, of those super resistant BI spores.
Which sounds bad. Why would partial kill prove anything?
Because if your process conditions are just barely killing, or maybe not even fully killing, the super tough BI, it demonstrates that those same conditions will absolutely annihilate the much weaker, naturally occurring bioburden.
Ah, okay. It's a relative comparison. Killing the BI is hard mode. Killing the bioburden is easy mode. So if you can handle hard mode even partially, easy mode is guaranteed.
That's the logic. It allows you to use gentler conditions that protect the product quality while still being completely confident you're achieving the required sterility assurance level for the actual bioburden.
That is clever. And you mentioned D values earlier.
Yeah.
How do they fit into this BBBI approach?
The D value is critical here. It's the time needed at a specific temperature, say 121 degrees C, to reduce the microbial population by 90%, or 1 log cycle. You need to know the D value of your BI in the actual product liquid, and you need a good estimate of the D value for your natural bioburden.
And the bioburden D value will be much smaller, right? They're easier to kill.
Typically, yes, much smaller. So the math, which involves the D values, the starting numbers of organisms, and the process lethality, F year, allows you to calculate the final probability of a non-sterile unit, PNSU. You can show that even a process barely capable of killing the BI achieves a PNSU way below 10 error for the bioburden.
The math proves the logic. Got it. So what are the specific steps in validating moist heat for liquids? It sounded like a lot.
It is quite detailed. First, you test the product and container, make sure they can actually withstand the heat without degrading or leaching impurities.
Step 1, don't break the product.
Right. Then, equipment qualification IQ, OQ, key Q, making sure the autoclave itself is installed right, operates correctly, and performs reliably.
And the temperature inside, you said that was extra critical for liquids.
Yes, empty chamber temperature distribution studies are done to ensure the chamber heats uniformly. For liquids, the temperature variation allowed across the chamber might be tighter than for items sterilized by direct steam contact, because you want every container to get a very similar heat treatment. BIs aren't usually needed for this empty chamber part.
Okay. What about the BIs when you do use them with liquids?
Careful selection again. They might be spores inoculated directly into vials of the actual product or self-contained BI units placed in the load. Crucially, you need to determine their D value in the specific liquid because the product formulation can affect their resistance.
How do you measure that D value accurately?
That must be done in a special, highly precise piece of lab equipment called a Biological Indicator Evaluation Resistometer, or BER. It delivers very precise, known lethal conditions.
A BRER. Okay. And inside the containers, do you put probes in?
Yes, for larger containers, say 100 milliliters or more, container mapping is done using temperature probes to find the internal cold spot or region, the place that heats up slowest. For smaller vials, mapping isn't usually feasible, so you rely on placement in the known coldest part of the chamber.
And how the containers are arranged matters too.
Definitely. Load positioning and mapping is key. You might need fixed loading patterns. Studies identify the coldest and hottest locations within a fully loaded chamber. Sometimes bracketing is used, validating the minimum and maximum load sizes to cover everything in between.
So what's the main event? The core validation study.
That's the heat penetration and microbiological challenge study. You place temperature probes and BIs together in the identified worst-case locations within the load, the cold spots in the containers, and the coolest part of the load pattern.
Bringing it all together.
Exactly. And the critical outcome is that the physical data, the measured temperature, the calculated FRO must correlate with the biological results, the kill rate of the BIs. If they don't match up, something's wrong, and you need to investigate.
And you also need to check the product quality afterwards.
Absolutely. Product quality and stability evaluation is ongoing. Does the product still meet all its specifications after being repeatedly sterilized under routine conditions? Appearance, potency, purity, container integrity, all checked. For lab media, you might check if it still promotes growth.
And finally, keeping it all working day to day.
That's routine process control. Formal procedures for everything. Regular calibration, monitoring the physical parameters every cycle, maybe monitoring bioburden levels periodically, strict change control if anything is modified, preventive maintenance, periodic reassessment of the validation, and of course ongoing personnel training. It's the system that ensures the validated state is maintained.
Wow. What an incredibly detailed picture. We've gone from the basic idea of sterility as a probability, explored all these different methods, steam, heat, radiation, gas, filters, aseptic processing, and really drilled down into the meticulous validation, especially for liquids.
It's a lot, isn't it? It really highlights that sterility isn't just about clean, it's about statistics, engineering, microbiology, and incredibly rigorous process control. That 1 in a million target requires a huge amount of scientific effort.
It really does. And it means that every time you, the listener, encounter a sterile product, whether it's an IV bag in a hospital or a simple eye drop bottle, there's this immense, unseen scientific undertaking behind it.
Yeah, this hidden dedication to precision, to managing probability, all to ensure safety in ways we rarely stop to think about.
So maybe the takeaway is just that appreciation for the hidden science. It makes you wonder what other everyday things rely on such complex, life-saving science that we just, take for granted. Something to think about.